Hello, my name is Josh Hill, and I'm an infectious disease physician at the Fred Hutchinson Cancer Research Center and the University of Washington in Seattle. And today I'd like to spend a few minutes talking about our new paper entitled Antibodies to Vaccine-Preventable Infections After CAR T-cell Therapy for B-cell Malignancies. These are my disclosures, which are all unrelated to this work. And so I thought I'd begin by talking a little bit about what a chimeric antigen receptor is. And so what this is a chimera of an antibody and a T-cell signaling sequence that is specific for whatever antigen of interest. And in this case, we're talking about a tumor antigen. And this CAR is expressed on the outside of a T-cell so that when engaged, it can trigger the T-cell to kill the target cell. And very briefly, T cells are taken out of an affected individual with a cancer. They are engineered in vitro to express the CAR on their surface, expanded in vitro, and then reinfused into the patient. And this has been used to target a number of hematologic malignancies, as well as now solid tumors, with very exciting response rates in these individuals. Now, it's important to understand that these are patients who have a high net state of immunosuppression. They all have refractory or relapsed cancer for the currently approved products. Many of them have received extensive prior therapies, many with a prior bone marrow transplant. In addition, before the CAR T cells are infused, these patients get lymphodepleting chemotherapy to help the CAR T cells expand in vivo. And as these CAR T cells expand and bind their target and are activated, they can release cytokines, which can result in the cytokine release syndrome or a cytokine storm and neurotoxicity, requiring additional treatments like steroids, which can further immunosuppression. And finally, these CAR T cells also destroy normal and healthy B cells in addition to the malignant cells. And so what this looks like is here you have your CD19 CAR T cell having its on-target, on-tumor effect killing malignant cells, but it can also have off-tumor effects, killing healthy, naive B cells. For CD19, we know that expression declines as these cells differentiate into long-lived plasma cells, which secrete antibodies. So plasma cells may actually escape this killing and be maintained. And so we hypothesized that previously acquired humoral immunity may be preserved in adults that get CD19 CAR T cell therapy, but perhaps not in other contexts, which I'll explain. And so here in the schematic, this individual gets CD19 CAR T cell therapy, and their yellow and orange or naive and memory B cells are killed, but we suspect these plasma cells will persist. And if that's the case, we hypothesize that whatever antibodies they have coming in will be maintained and stable over time, even after successful CAR T cell therapy. However, if this dashed line is normal diversity, because of their prior therapies, these patients probably have gaps in their pre-existing antibody titers. In kids, we think that perhaps their pre-existing plasma cell clones are fewer in number and less mature. So although whatever they have to start with as far as antibodies may be maintained, their set point or diversity may be lower. But in adults getting BCMA CAR T cell therapy for multiple myeloma, these CAR T cells specifically target plasma cells. So you have the opposite scenario in which all plasma cells are destroyed. And in this case, we hypothesize that antibody titers will actually decline rapidly after treatment. So to explore these questions, we performed a prospective observational cross-sectional study of adults and kids getting CD19 or BCMA CAR T cell therapy. We enrolled individuals who were at least 6 months or more out from getting CAR T cell therapy and in remission, so they were successfully cured of their malignancy. And we obtained blood at one time point from these patients, and we tested their blood for antibodies to 12 vaccine-preventable pathogens indicated here. And we also did this serosurvey using the VIR-scan assay, which is a bacteriophage display to identify antibodies targeting any viral epitopes or a select number of bacterial epitopes to understand more comprehensively and globally what types of antibodies these individuals have in their blood. So we recruited 54 CD19 CAR T cell therapy recipients in remission and 11 BCMA CAR T cell therapy recipients. There were a few kids, but mainly adults, and about half of these individuals had received immunoglobulin replacement therapy for low total immunoglobulin levels. And those individuals were were described separately as necessary for specific analyses. So in this summary slide, which really shows the majority of what we found, on the x-axis here, I'm showing each of the specific pathogens that we tested antibodies for. And on the y-axis, I'm showing the proportion of individuals that had seroprotective antibody titers. And in the dark blue group here are those individuals who had not gotten any immunoglobulin replacement therapy. So there's no confounding by antibodies that are infused as part of their prophylactic treatments. And when you compare that to the gray bars, these are data that come from serosurveys of healthy individuals in the US, or population-based studies in the US. What we see is that for just about every pathogen, the dark blue compared to the gray blue is relatively similar, indicating that these CAR T-cell therapy recipients in remission actually have similar antibody protection as compared to the general population. What you see in the light blue are individuals who had recently received immunoglobulin replacement therapy in the prior 16 weeks in the last few months. And as expected, you can see this does boost up their antibody, the number of patients that have seroprotective antibody titers, particularly for some of these other pathogens like hepatitis A, B, and streptococcus pneumonia. When we separated this out by CD19 versus BCMA CAR T-cell recipients, we see some signs that suggest our hypotheses were correct, in that BCMA CAR T-cell therapy recipients, on average, had fewer numbers of people that were seroprotected compared to CD19 CAR T-cell therapy recipients. There was even one BCMA CAR T-cell therapy recipient who had no seroprotective titers to any of these 12 tested pathogens, really driving home the point that these patients getting BCMA CAR T-cell therapies have very compromised humoral immunity. We also looked in some generalized estimating models at whether or not, on average, patients in each of these categories have more or less seroprotective antibody titers, and we see that on average, patients who received BCMA CAR T-cell therapy had fewer seroprotective titers compared to CD19, did not reach statistical significance, but we do note that there were only 4 or 5 individuals in this BCMA group. But very interestingly, the total IgG level above or below 400 milligrams per deciliter did not correlate at all with whether or not someone had seroprotective titers. So this does not seem to be a good correlate or surrogate for pathogen-specific antibody titers. Similarly, for CD19 positive B cell counts in the blood, we didn't see any clear association with lower or higher counts and seroprotective antibody titers. We also used the VIR-SCAN assay to look more globally at the absolute number of epitope hits recognized by antibodies in an individual's serum samples. And again, we see that individuals who got BCMA CAR T-cell therapy had significantly fewer antibodies to viral or bacterial epitopes compared to CD19 CAR T-cell therapy recipients. And we see a similar pattern for patients who previously received hematopoietic cell transplants. So in summary, seroprotection for vaccine-preventable infections in adult CD19 CAR T-cell therapy recipients was comparable to the general population. I say adult because pediatric patients all had received immunoglobulin replacement therapy in the study, so we couldn't make conclusions about that group. And the BCMA CAR T-cell therapy recipients appear to have fewer pathogen-specific antibodies, but both of these groups have important deficits in specific pathogens like strep pneumo. So clearly there's a need to understand vaccine efficacy in these patients, as well as the clinical benefit of immunoglobulin replacement therapy. And lots more questions to learn, and we look forward to doing more studies to further understand these questions in this patient population.